Thank you very much. And first of all, I would like to thank ILSI for providing me with this opportunity to take part in this major project and to also share with you some of the work that we have done on viruses in this project. And I also would like to thank Dr. Farber, who used to be with us before, and I thought when he left that we have sort of a kind of a relief period, and all of a sudden the phone rings and he says, we need you on virology now. So it was a great initiative from Jeff, and I appreciate that very much. Thank you again. One thing I'd like to comment on is the quantities of foods that we have received. And of course, as Jeff mentioned, there were buckets and buckets of cereals and pistachios, but the most interesting was the buckets of chocolate. What was disappointing a little bit, it was not a Swiss chocolate, but it was fine for our purposes. The reason we are working on viruses is because our laboratory in Health Canada is the National Reference Lab for Food Virology. And we do a lot of work with method development, as well as on studies on inactivation, transfer of viruses from hands or food handlers to foods, as well as moving into the whole genome sequencing events at this point in time. As you know, there are two major viruses that have been involved or implicated in major outbreaks with respect to illnesses. And these two viruses are norovirus and hepatitis A virus. Very briefly, just as a refresher, norovirus is the leading cause of non-bacterial gastroenteritis agent. It afflicts millions of people with many hospitalizations, and it is now among the major. It costs about 40% of the infections are because of foodborne origin. The transmission is through foods, contaminated foods, person to person, through environment, through water. And the interesting thing about norovirus, it's a very low infectious dose. It's a highly infectious organism that as low as one virus particle, it's been reported, could lead into infection. And the incubation period is just about a few hours to less than 2 days, and then you end up with this explosive characteristics of projectile vomiting and severe diarrhea associated with abdominal cramps. The problem with this virus is, as infectious as it is, as important as it may be, we still do not have a robust cell culture to amplify this virus, to grow it, to study its characteristics. So we were very limited. We continue to be limited in our attempts to target the virus itself. For this purpose, and for the purpose of this study, we resorted to using surrogate viruses, mostly the feline calicivirus and the murine norovirus. And these are very well and well characterized viruses as surrogates. Each has its own pluses and minuses, so we brought both of them. In this way, it will give us an overall picture of the behavior of norovirus in foods. Hepatitis A, on the other hand, causes acute liver disease, and it is also transmitted through foods, water, blood, or human contact due to different lifestyles. Again, the infectious dose can be low, about 1 to 10 virus particles. The unique things I think about hepatitis A is the incubation period, which spans up to about 50 days, an average about 28 to 30 days, by which time, of course, the implicated or suspected food has disappeared. So you cannot really go back and look for it in the food. It is environmentally stable, just like norovirus. Both viruses are very stable in the environment, and they can survive for weeks and months without much change. But with hepatitis A, we do have a cell culture, but for all practical purposes, the cell culture is used mostly with lab-adapted strain of hepatitis A. So we use these viruses, the surrogates and hepatitis A, in our studies. Now, something to also remember that unlike bacteria, and these are challenges that virologists have all the time, these viruses do not grow on foods, do not multiply. If you have 10 particles, you end up with 10 particles. So the challenge is to extract those 10 particles, because the amount of virus that infects or contaminates foods is also reasonably low. There's no culture media that you can amplify the virus as well. So you are so limited with your ability and technologies to retrieve those viruses in an intact form and in a good number to allow you to detect by the detection limit methodologies we have available to us. These viruses have been involved in many outbreaks. I've listed many here, but we're not going to go through these. And in different settings and with different foods. So we have restaurants, homes, nurseries, senior homes. Cruise ships is an important area too. And a variety of foods, from shellfish to salads to cakes to strawberries. There are reported few reports on the association of dried food, low moisture dried foods, and these viruses. And for the purposes of this meeting, I decided to just list two of these, just to give us an idea. So one of them is there were 7 foodborne outbreaks of norovirus in Japan, where more than 2,000 people were ill because of a product called nori, which is a dried shredded seaweed. In Europe, there was a hepatitis A outbreak associated with semi-dried tomato, and it was in England and Netherlands, and then it spread also to Germany and other countries. So this gives us an idea that these viruses are there. They can contribute to illness if these products are contaminated. The question we have is, how long these viruses survive? How can you inactivate these viruses if we find them there? Any changes happening in these viruses genetically over a long period of incubation? And what methods we need to use to extract these viruses from these foods? So the objectives of these, so these are the rationales for why we are conducting this study as part of this ILSI project, is because LMF, low moisture foods, are usually considered as ready-to-eat products. So you don't have a heating step before you consume the product. So you're eating the product with the virus live if it's there. And because of the association of some of these foods with these outbreaks, as well as the lack of data of the survival and inactivation, this project made sense to us. So the objectives was to develop or to use whatever methods available now, and this is also a challenge that we have in virology, unlike bacteriology. Our methodology is quite limited. There are many methods, but they're not very efficient for virus recovery. And that's our major problem, is to recover the virus from the product. So we try to use whatever available method that makes sense to us and try to improve on some that we have already started to work on in our laboratory in the past. And also to investigate the survival of these viruses in these foods, and then to assess a new technology called advanced oxidative process to inactivate these viruses in foods, and I'll explain all this when we come to it. And then to do whole genome sequencing, a deep whole genome sequencing, to assess if there is any change happening in the genomic makeup of the viruses over the period of incubation or storage in these products. So the methodology, basically, we chose to work with the pistachios, chocolate, and corn flakes. And we used the hepatitis A virus, lab-adapted strain, of course, and the 2 surrogates for norovirus. And basically, we inoculated these commodities with 100 microliter of stock culture of 10 to the 7 plaque forming units per ml into 25 grams of these products. Then we incubated these products for a period of 24 hours, 1 week, and up to about 4 weeks. at 22, 23 degrees plus minus, depending on the environment. And then we sampled at each of these intervals to look to determine if the virus is surviving, how much we're losing of the virus, if any, or not. And we use for this purpose two methods. So there is an ISO methodology, which is the 15216. And this is an overall method used for virus, and it's validated. But it's not always very, very efficient. But this is what we have at present. That is a standardized, validated method. But at the same time, we also used a method that we have developed, which is the porcine gastric mucin-coated beads to recover the virus. So using the ISO method steps to recover the virus and using this method, we use them in parallel. And then once we recover the virus by either of these methods, then we try to quantitate the virus by using either the plaque assay, traditional plaque assay, or by using the digital droplet PCR, either through this mechanism or through the ISO mechanism methodology. The ddPCR, we have been working with this for at least 2 years now, and all the data that we have show that this methodology is in fact better than the ISO method in capturing the genomic copies when it is compared to the ISO method. So, and there are reports in the literature, recent reports from France as well, to support this notion. So we use this method to recover the virus. The results are, because we're using 3 different types of viruses, we're using different methodologies, different commodities, it can be very confusing. So I try to sort of minimize the confusion by putting 2 slides per each category, just to give you an idea about the trend of what we have seen so far. This is still a work in progress, of course. So when we look at the results for the feline calicium virus survival on these foods, on pistachio and chocolate, we find that we have barely lost, and this is done by the digital PCR, of course, that's the copy numbers of the genomes. We see that we barely lost about 0.5 of a log over a 4-week period. When we looked at the same commodities using the plaque assay, which is a quantitative visual mechanism, we see that the infectious virus was lost by about 1 log or less. So we have slightly more of a loss of the infectious virus as compared to its genetic material over a 4-week period. When we look at the Corn Flakes, we also see that we only lost about 0.5, 0.6 of a log. Our plaque assay is still in progress, so I cannot report on this at this point in time. When we look at the results for the MNV virus, we see similar trend. We see just about 0.7 to 0.6 log loss over a 4-week period on chocolate and pistachios. That is in the genomic copy number. And when we compare this to the plaque assay, that is for the infectious virus, we find that, again, similar. We're losing just about 1 log, 1.1 log over a 4-week period. Again, for the Corn Flakes, the picture is similar. It ranges around 0.5 log reduction. So in summary, for these 2 viruses, which represent norovirus, genomic copy number loss is about 0.5 log. The actual virus, infectious virus loss is about 1 log over a 4-week period. For hepatitis A, it's basically the same picture. We were surprised. It's the same picture. We have, again, almost a 0.5 loss of a log. And on Corn Flakes, with the plaque assay, we had actually about just 0.2% 2.2 log reduction in the infectivity of the virus. So whether this tells us that whether this is significant or not, I don't think so. But if it is, it tells us that this virus could be more resistant in this, or survives better in this kind of commodity. What we did next is, okay, this is still continuing, of course. These are just results at this point in time. What we decided to do is to see, have there been any changes in the genomic makeup of these viruses over a 4-week period? The objective is to see if the interaction, virus-host interaction with this particular type of food is causing the genome any changes. Now, what we did is SNP analysis, of course, by deep whole genome sequencing, not just whole genome sequencing, but deep whole genome sequencing. So we zoomed onto the ORF1, 2, and 3, and we found that over the 4-week period, we had changes in these positions in ORF1, changes in these positions, and this is the same position that seems to a different population to show different changes, but none in ORF3. Keep in mind that this is the one that is under the highest pressure because this is the capsid part of the virus, as opposed to the polymerase also. But this is exposed to the environmental pressure probably more than this ORF. Now, is this significant or not? We can discuss this as a question if you like afterwards. Now, then we did also some inactivation studies by advanced oxidative process. And this is an equipment developed in Canada, I believe in Guelph. Jeff, am I correct? Or somewhere there. We are partnering with these manufacturers to test this equipment, which uses a combination of ozone, UVC, as well as peroxide, hydrogen peroxide. All combined in one exposure parameter. I cannot, at this point in time, tell you what the concentrations are. This is still a proprietary equipment and technology. So, but basically, the equipment is here, and basically what you do is inside this chamber, there is a conveyor belt, and you put your sample here, and the belt moves at a specific time, speed. We sampled at 30 seconds and 60 seconds. So at 30 seconds is here, and we took the sample out. And at 60 seconds, we took the sample at this point to determine if exposure of the virus to this process caused any reduction in the virus titer or not. We were really pleasantly surprised to find that, and I've converted this to percentage just to give you a better idea rather than log. And basically, we found that on these commodities, we had a minimum of 85, we had at 60 seconds, we had a minimum of 85% to almost 100%. It's 98.9%. Inactivation of the virus on these products after 60-second exposure. This is by the plaque assay. So when we're looking at the infectious virus itself. When we did that with the digital PCR, we found that we have less reduction in the genomic copies as compared to the infectious virus. So we had about 27, 28% with respect to a reduction on the corn flakes, and we had just about 80% or so when it is on chocolate and pistachios. So we can see that in both situations there is a greater reduction on these products using that machine, that equipment, as compared to the corn flakes. And with the corn flakes, the genome reduction is much less compared to the reduction in the infectious virus itself. So I would like to just conclude here by saying that, of course, this is still a work in progress. And we found that the magnetic bead-based method that we developed in our laboratory is comparable, produce comparable recoveries. Again, this is a challenge. And when we say 5 to 50, it's massive range. But this is very normal for virus recoveries. If you work in this field, you know you could recover only 1% sometimes. and sometimes you recover 60%, depending on how lucky you are on that day and the different types of technologies you're using. But we fall within the normal range of virus recovery, and both produce the same recoveries. The difference is the method that we've developed is simpler and faster to use. It takes 1 hour versus 5 hours as compared to the ISO method. The bead-based method did not work for Corn Flakes, and we can discuss this afterwards if you wish. The survival of the genome, we only found or observed minimal decay, as I mentioned, in the genome, in the decay of the genomes of FCV, MMV, and HAV in any of the foods tested during the 4-week incubation period. And when we look at the survival of the virus, again, the infectivity, we have only a reduction of about 1 or less than a 1 log reduction of the virus over the 4-week period incubation on pistachios and chocolates. When we looked at the inactivation of these viruses by the AOP, nearly 85% to 100% reduction in viral infectivity, and I emphasize infectivity. This is by the plaque assay observed with the AOP when we expose it for 1 minute. With whole genome sequencing, we were able to, deep sequencing, we were able to identify certain SNPs that have changed from time 0 to time of 4 weeks. We are continuing this work, of course. In fact, as I was coming here, I just got an email from the people back home saying, we have a new graph. I said, it's too late, I can't put that on. So maybe that's for next IAFP. So we have inactivation studies are continuing. Infectivity assays will be completed hopefully by the end of this year. Similarly, the virus recovery, HAV, in pistachios and chocolates and cornflakes will also be completed. And we will, of course, what we saw with the whole genome sequencing is just an initial input that we have seen so far. So we're continuing this work to see if this is a continuing trend or is just by chance. With this, I would like to acknowledge these people who did the work, and I wish they were here so they can answer your questions, but I'll be happy to answer some of your questions. And with that, I'd like to thank you for listening to me. Thank you.
Thank you, Dr. Bidawid. Exciting work. The more you know, the more questions come up, and the more you scratch your head. But this work's not done yet, so stay tuned. Questions? Anybody? Uh-oh.
I was counting to 5 and said maybe nobody.
Brad Marks from Michigan State. I'm curious your opinion, your interpretation of the AOP results, because on one side you say I store it for a month and I have less than a log degradation.
Yes.
But then you applied this treatment and really you got less than a log of reduction.
Yes.
You're talking about 85. So in a sense, storing in a month is the same as applying that treatment.
Correct.
Roughly.
Correct.
And so mechanistically, do you think this AOP is really effective in doing anything on the virus?
Absolutely, you're very right, and that's why I put it in percentage, just to give you the idea that, okay, we can speak of percentages, but it doesn't mean that we're removing the whole virus. Right. So we're removing only about 1 log, 1.5 log from that commodity. The question now is, do we need to play around with the concentrations of these components in this machine or the time frame? This is just still in its developing stages, but we find it to be very easy to use. And especially, we're trying to develop a mechanism where if you use, let's say, strawberries, the machine will then automatically tumble the stuff so you expose the whole surface, not just the one surface. So this is being worked with as we are speaking now, and this is something that we have thought about. But I agree with you. It is, yeah, it's about a 1.5 reduction.
Thanks.
Oh, looks like there's one more. You don't get off so easy.
That's right.
A quick question about the mutations that you see. How can one differentiate whether they arose during that time versus that they were pre-existing in the original viral inoculum?
Right, and I anticipated that question. That's why I left it to the end. So, and this is a question that we have discussed also in our lab. So as you know, RNA viruses especially mutate a lot. And there's always a mixture of these viruses that may have different SNPs at the same time. But when you do, this is by regular whole genome sequencing, but when you do deep sequencing, you're zooming on particular elements of that genome. We have seen that some of these have only shown up when after the 4 weeks, at the end of the 4-week incubation in these foods, but they were not there at the beginning at time 0. The question is still there. Is that because, is it a change in the population that's happening? Or is it because these viruses that have shown these changes have responded to the effect of the dryness of these foods and did something to make them live better or survive better? A question still open to a discussion, but until we see the trend, I cannot give you a definite answer at this point in time.
All right. Nice round of applause for Dr. Bidawid.
Thank you.